Okay, good afternoon, everybody. And we're going to carry on our discussion about viruses. First of all, we will complete our general discussion about viral biology, and then I will do a class on viral disease. I'm not sure how far we'll get with this, but I want to finish it. I want to finish this week, so I may put in an extra class, an extra recorded shorter class, just complete viruses before we move on to talking about bacteria. So we have talked in general about the structure of viruses. Now we need to start thinking about the fact that there are many different kinds of virus. All living things have their own viruses that infect them, and viruses can cause a very wide range of effects, especially in disease-causing viruses. We recognize particular virus disease very often as a syndrome, as a collection of symptoms which are often repeated for that particular virus. So symptomology is very important in identifying what virus we're actually dealing with, particularly when we're looking at somebody who's ill. The second thing that we can do is experimentally, we can begin to look at particular cytopathic effects directly on cells, remembering that viruses infect particular cells. They are very particular, they are very specific to certain kinds of cells in certain hosts. And if we can look at effects of a virus on the cells themselves, we can often get close to identifying what virus it is that we are looking at. Very widely in clinical practice, serological tests are used to identify virus. That is, that we use specific antibodies, specific identified antibodies, to react with virus experimentally, with samples, for example, of serum or whatever. We can mix them with antibodies which we've derived from some source. And if remembering that antibody is exquisitely specific to a particular antigen, we can react particular antibodies with the virus and actually see an effect and be able to identify the virus that way. But increasingly in clinical settings, what is being done is we are actually identifying not by trying to react with the outside of the virus, which is what most of the serological tests do, but instead to look at the nucleic acid. All viruses, that is the one thing which all viruses do possess, and that is a nucleic acid genome, either DNA or RNA, depends on the virus. But we can actually extract that from samples and identify it. Often it is amplified in polymerase chain reaction, which we discussed before, and we can identify nucleic acid. And not only that, we can see how much nucleic acid there is present in the infected person or the infected animal, if we're dealing with an experimental animal. We can actually see a degree of illness by the amount of virus that is present in the blood, for example. Of two instances of that I will mention. The first is that the PCR, polymerase chain reaction amplification of RNA, is now a standard way of identifying COVID-19 infection. And many of you who have been tested for the virus, that is exactly the test that they would have done. They would have amplified any RNA present in the sample by PCR and then isolated and identified it. The second instance that I would mention is in connection with HIV infection. in human immunodeficiency virus infection, the level of virus is very closely monitored. And it is done by PCR, by polymerase chain reaction. In most cases, in many, many cases now, where people are treated for HIV infection, that viral RNA level is brought to 0, which is quite remarkable. Unfortunately, it does not stay at 0 if the drug treatment is lifted. And with drug treatment, the RNA levels are brought to 0. Effectively, there is no virus kept in circulation in the blood, which is a very good thing because it means that people with that lower viral burden aren't infectious, nor will they suffer from the disease associated with the syndrome. Okay, so the viral identification is extremely complicated, and there's lots and lots and lots of scientific fighting about how exactly we should identify viruses, how we should classify viruses. None of our classical identification schemes apply to viruses because they are not living things. Most other taxonomy of bacteria, of animals, plants, whatever, the taxonomy reflects some sort of evolutionary relationship, which allows us to organize what we're looking at. Viruses, we don't see that at all. We don't see that one virus is, you know, ancestral to another, etc., etc. So it becomes very, very difficult to classify viruses. But we do so in general on 2 features. The first is the nature of the genome, whether it is RNA, DNA, and you'll see that in a minute. That has complications as well. Second of all, whether or not there are enveloped viruses or not. Those are 2 primary ways that we'll see that they start to organize. This is done by an international committee on the taxonomy of viruses. Few of the members of that committee actually speak to any of the other committee members because they're all so busy fighting about exactly how these schemes should work. But we're just going to take a simple look at it. And my idea here is not that you should memorize each of these, but that you should be aware of the fact that this is a very complicated topic. And so we'll have a look. First of all, I have already mentioned that viruses can be divided into 2 groups, and these are the RNA viruses and the DNA viruses. So let's have a look first at some of the DNA viruses. And DNA viruses, they all have DNA in one form or another as the genome held inside a capsid. And many of them are double-stranded DNA. Well, that's perfectly natural because DNA is inherently a double-stranded molecule. It's that beautiful double helix. So that shouldn't surprise us. The DNA viruses have this group have double-stranded DNA. But there are those which are enveloped. That is, they have an envelope around the capsid, which is derived from some host membrane. And then there are those which are unenveloped. Now, well, let's just have a look. So we've got DNA viruses, double-stranded, either enveloped or unenveloped. Okay, let's have a look at, I'll mention particularly important ones. First of all, this group here, the adenoviridae, the adenoviruses. Now, adenoviruses are very, very diverse. This is only a small grouping of them. But one thing that we do commonly encounter them, they are one of 200 or more viruses that cause symptoms of the common cold. The other thing that I'll mention to you just out of interest, adenoviruses are often used, genetically manipulated as a viral delivery method for genetic engineering. And they have been used in humans. What they do is they're non-enveloped. So they just have a capsid. They have a geometric shaped capsid. And they construct artificially, they get empty adenovirus capsules, and they pack DNA into it that is useful. And then the outside of the virus remains infectious. But when it infects a cell, it delivers the DNA of interest, or the nucleic acid of interest. It doesn't have to be DNA when you're doing it artificially. And I mention this because one of the current viral COVID mechanisms actually does this. It uses an adenovirus as a delivery vehicle. Adenoviruses, otherwise, they cause a wide variety of diseases, most of them mild, but they can cause quite a serious viral meningitis. It's not nearly as dangerous as the bacterial meningitis that we'll hear about later, or that I've mentioned before. Viral meningitis tends to be much milder. The other group of non-enveloped double-stranded DNA viruses that we really must think about, though, are these, the Papilloma Viruses. These are the Papilloma Viruses. They are best known as viruses which cause warts of one sort or another. So they're very, very common, extremely widespread. There are a number of different strains of Papilloma Viruses. But were of particular interest for us because they not only cause warts, but they are able to transform cells into tumor-forming cells. So they are oncogenic. They are able to spark the development of cancers. And the one group that we're particularly interested in are the papilloma viruses that cause genital warts. Genital warts are a very, very common, very easily communicated viral infection. Normally, they simply cause warts around the genital region, and they are treated, they're usually treated the way you treat any warts. They get burned off with liquid nitrogen, whatever, and usually in most people eventually develop a resistance, and they will disappear. The problem is they don't really disappear. These are one of the groups of viruses which can hang around in a latent form. And what the papilloma viruses do is they cause cancers. They are a common cause of cervical cancer in women. And they can also cause cancer of the penis in men. That is not common overall, but where it does occur, papillomavirus is a common cause of penile cancer. But penile cancer overall is rather rare. Cervical cancer is not. Cervical cancer is relatively common. There is now a very good vaccination, vaccine series. You have at least two injections for papillomavirus. And I will talk more about it later on, about the complications associated. But in any case, there's a very good vaccine against papillomavirus, which prevents both genital warts and its associated cancers. The advice is that this be given before puberty and before any sexual activity begins. Okay. All right, so those are non-enveloped. This is just a few of them, by the way. There are others. Also double-stranded, but enveloped. Here are some very important ones, especially in terms of human illness. The first are the pox viruses. And the most important one in human beings was smallpox. Smallpox is a virulent disease with a high mortality, which can cause severe disfigurement caused by an enveloped double-stranded DNA virus. This virus now no longer exists, excepting in laboratory collections in 1 or 2 laboratories around the world. And this virus was eliminated from the human population by a vigorous vaccination program. and it was declared dead in 1976. So those are the pox viruses. There are a number of other pox viruses which occur in other animals, and one of them, cowpox virus, was one that was used to actually vaccinate human beings. The other group of enveloped double-stranded DNA viruses that are tremendously important are the herpes viruses. Here, they cause a very wide range of illnesses in human beings. Cold sores are the most benign. Cold sores, genital herpes, chicken pox, which is rather more serious, and shingles, which is caused by the same virus as causes chicken pox. The herpes viruses all have this tendency to become latent. Very often, in most of these ones here, cold sores, chickenpox, shingles, they actually retreat to nerve cells where they remain latent. They may remain latent for your entire life and never outbreak, but they can do so. That you can have recurrences. The other one, the other enveloped one, I'm not going to talk about in any detail. A very peculiar virus called the Hepatonoviruses that cause hepatitis B, but don't worry about these. We won't. I may mention them again when we talk about illnesses, but they're rather strange. But here's something really peculiar. Viruses often pop up these really strange ways of reproducing, and this group of viruses that are single-stranded DNA. It's very, very peculiar. It's just one strand. It's what we would regard as being the template strand if you were talking about transcription, translation, etc. They have what is the equivalent of the template strand with the genes on it, but it's just a single strand, and they are non-enveloped. They just have the DNA and then the capsid. And those of you who have dogs may have heard of this group of viruses, the Parvoviridae, which they can cause in humans, they can cause a gastroenteritis, but in animals, they can produce a very, very serious group of illnesses. Very strange, single-stranded and non-enveloped. I'll also mention that what they have not included here are a very large group of DNA viruses, which are bacteriophages, which we'll mention in a minute. These are only viruses. The viruses here are only those which concern human beings and other vertebrates. Okay. In terms of human disease, the RNA viruses are much more important than DNA viruses in that, from the point of view that they are much more diverse. Many more illnesses are caused by RNA viruses than by the DNA viruses. And the RNA viruses, well, there's something that I need to explain, and that is they can have single-stranded RNA, which many of them do, most of them do. They have single-stranded RNA as their genome. But they may have DNA, and then, double-stranded RNA as the genome. Very peculiar, but not so difficult to visualize, because you'll remember that RNA can actually, even in a normal cell, RNA can fold back on itself, although it's single-stranded, it can fold back on itself, and its base pairs can match up together. That's how we fold RNA into a structure in the cell. So RNA can base pair to itself. It just doesn't normally do it as a double strand. But here in these viruses, it does. It's a double-stranded RNA. They are non-enveloped. And this is a group of viruses called the Rheo Viruses. And they are best known for causing things like gastroenteritis. Just remember that we have them. The double-stranded RNA, which are non-enveloped. We need to spend far more time talking about the single-stranded RNAs, because we're going to hear about many, many important disease-causing viruses in this group. Now, single-stranded RNA, there are two kinds. The first is called positive strand RNA. That's single-stranded RNA that is essentially exactly like messenger RNA. When it ends up in the cytoplasm of a cell, it can just be read off by ribosomes immediately. It's just like messenger RNA. The other group have negative strand RNA, negative single-stranded RNA, and this needs to be copied. So you have single-stranded negative RNA. You need a copy of it that will then function as messenger RNA. These require another step in order to be replicated. So we'll talk about the positive strand first. It's the easiest to understand. Single-stranded RNA is just like messenger RNA, ends up in the cell, gets replicated, and it gets transcribed, it gets translated rather, et cetera, et cetera, et cetera. So positive strand, non-enveloped. There are many, many, many of them. But for example, this group called the Papilloma Viruses, many of our common cold viruses are in the Papilloma Viruses, but there are some very, very dangerous viruses here as well. Polio, for example, is caused by a Papilloma virus, a positive strand RNA virus, which is unenveloped. There's another virus which causes hepatitis A. Hepatitis A is the common hepatitis, often called yellow jaundice, a relatively benign form of hepatitis with a duration of some weeks of inflammation of the liver, which usually settles with no sequelae at all. It is not like hepatitis B, hepatitis C, which are extremely serious and with which have long-term consequences. Never mind about the calicivirus. Just remember Papilloma Viruses there. So positive strand enveloped viruses. Ooh, here now we start to hear some familiar names for us. First of all, here, this group here, the Toga Viruses, that cause this disease here called rubella, also sometimes called German measles, and it's a measles virus. It's not a measles virus, it produces measles symptoms in that you get little red spots. It's a relatively mild disease, rubella. It's included in childhood vaccinations, however, for one very important reason, and that is that rubella in pregnant women is extremely serious and causes developmental defects in the fetus. The disease itself is usually extremely mild, even in children and whatever, but in pregnant women, it has this disastrous effect that it can cause birth defects. This group here, the Flaviviruses, flavi means yellow, and they're easy to remember because one of the diseases that they cause is yellow fever. They cause this whole group of fevers here: yellow fever, dengue fever, West Nile fever, which have similar symptoms. And we are somewhat interested in them because these West Nile fever, dengue fever, we are at risk of seeing these viruses spread from the tropical areas where they now occur in southern states, further up, particularly with global climate change and with changing range of their vector, which is the mosquito. So these are transmitted by mosquito. The next group, of course, are the ones that we are absolutely enthralled with at the moment because we are in the midst of a global pandemic caused by one of them. And these are the coronaviruses. So the coronaviruses are enveloped, positive strand, single-strand RNA viruses. Important to remember. This whole group of the coronaviruses, in human beings, 4 very well-known ones, which cause a mild illness, which is like the common cold. They cause symptoms of the common cold. Those we've known about for many decades. But other ones are recent entries to the human population. The first major one was SARS, severe acute respiratory syndrome. That was SARS CoV-1. And SARS CoV-1 caused a very, very serious illness with a high mortality rate. But the disease produced symptoms very quickly, and people became very ill very quickly, so they did not spread the disease very widely. SARS CoV-2, our current coronavirus, is much more dangerous in many ways. It has a lower mortality rate, but people with the disease can spread it for far longer without realizing that they are ill. It is also very, very contagious. It's much more contagious than SARS CoV-1. There are some other diseases which we'll hear about later associated with this group. The other group, which is of tremendous importance still, are the retroviridie. Now, these viruses are a little bit different. All of these viruses here and these ones here, they just use their single-stranded RNA as the hereditary material as it is. They reproduce that RNA for the new genomes. They have one or other strategy for reproducing them. They just produce RNA from the RNA, in other words. But this group here, called the retroviridie, the backwards viruses, in other words. The retroviridie are those viruses like human immunodeficiency virus, and they have a separate system. And that is that they take this this equivalent of messenger RNA strand, the positive strand, and they produce a DNA strand from it. Now, that is the reverse of what we're used to. We're used to hearing that DNA is transcribed to messenger RNA, which is translated to the proteins, right? Here we've got RNA, which is being reverse transcribed. RNA is now being reverse transcribed to a DNA strand. And that DNA strand then becomes part of the host genome and eventually may go on to producing masses of HIV virus. There are several of these retroviridie, but the only one which causes really commonly causes serious illness in humans is human immunodeficiency virus. There are a number of others which are known in animals, but we will talk a little bit about HIV because this is the causative agent of AIDS, acquired immunodeficiency syndrome. Okay, so single-stranded RNA. Now let's have a look at the negative strand, remembering that the negative strand RNA viruses, they need to convert this negative strand, they copy it into a positive strand. So they undergo an additional step. And we'll only talk about enveloped viruses, enveloped negative strand viruses. But here you'll hear some important ones. The filoviridie, philo means like a strand, a thread. And you'll remember I told you Ebola looks like that. Ebola is a long thread-like virus with looks like a carpet beater at the end, a loop at the end. This whole group, Ebola, Marburg, these are all hemorrhagic fevers, very virulent fevers, which produce this widespread hemorrhaging, particularly from mucous membranes, with a very high mortality. Ebola, Marburg, there are numerous other hemorrhagic fevers, all of them associated with this group, the filoviridie. Never mind about the bunyaviridie for the moment. This group here, I also want to mention the rabidoviridie. This is rabies. Rabies is a neurological disease. It is widespread in mammals. It's a promiscuous virus. It can infect many different hosts as long as they are mammals. And it's a dreadful disease. Once it is symptomatic, it has an incredibly high mortality rate. The mortality rate is 99%, basically, once the disease becomes symptomatic. But it is a slow-moving disease. So there are interventions that you can undertake. If you know if somebody has been infected, in fact, vaccinating them works because you can get the immune system to work quicker than the virus establishes its infection. It's a very slow-moving virus, but once it becomes symptomatic, it's practically 100% fatal. Orthomyxovirus, that's one you must remember. Orthomyxovirus, these are the influenzas, and we'll talk some more about them. Paramyxovirus, very important childhood disease causing agents. These are the cause of mumps and of measles. Those are both still extremely important childhood diseases. People do kind of belittle these childhood diseases and say, well, they're nothing. If children get them, then they carry a very high mortality. And they also have a strong tendency to produce dangerous sequelae after they have passed. So these diseases are extremely serious diseases. And mumps, the vaccinations against mumps, measles, and rubella are usually given together as the MMR vaccine. Never mind about Lassa fever. Okay, so that gives you an idea of the huge diversity of these viruses. DNA, double-stranded or single-stranded, enveloped, non-enveloped, RNA, single-stranded, positive strand, negative strand, even double-stranded RNA. Very, very diverse, some of them quite peculiar in the way that they reproduce. Okay, so when we look at the growth of viruses in a host, we don't see the same sorts of growth patterns that we saw in the bacteria. And you remember in the bacteria, we had a lag phase, then an exponential growth phase. Then as nutrients and everything or whatever happened, the growth leveled off and then the population would decline. And very often in bacterial illness, that the growth curve basically also describes the course of the illness. But viral multiplication is a little bit different. Viruses very often will infect, provided you acquire an infectious dose. They have to invade cells. They have to take over the cell machinery and get the cell to basically become a viral factory and produce viral particles. And those virus particles very often are released in a burst. That burst may last sometimes. Sometimes it doesn't, as we'll see. But it's not, the cell does not carry on producing virus indefinitely. The production of virus is very damaging to the cell, and the cell will eventually die. So what we observe is the following. If we follow the viruses, the number of viruses, we were tracking how many viruses we could see in circulation, for example. What we would see an initial circulation of virus after infection, but then the number of viruses that we see simply disappears. What has happened is they've all got infected their requisite cells. Viruses which are in circulation may have been eliminated by the immune system. The immune system may be responding to them already. But a huge number of viruses now have gone into cells and have retreated there for a time, turning the cells into this viral factory. And then all of a sudden, they all start to get released. They get released in a burst. The number of viral particles increases geometrically, not exponentially, it increases geometrically. The viruses are released, go out then to infect more of the host cells. So we see a very rapid increase in the number of viruses in circulation. And this is a period of acute infection. And in this time here, hopefully, the host immune system is responding. The host immune system will respond until such time as it begins to eliminate the virus, and then the number of viral particles in circulation will drop. So we see this burst and then a sudden decrease. This period usually described in terms of days or weeks for most viral infections. So we have a period, we might regard this as being the prodromal period, when virus is present, but we are asymptomatic. Then as virus is released, we become symptomatic from the damage the virus is doing, but then we also pass into a period where we accumulate symptoms also from the action of the immune system. And it's during this period of acute infection here that we probably feel most ill, especially towards the end here as the virus begins to get eliminated. Then we pass into the period of convalescence. So again, we can track the course of illness with the amount of virus present in the body, but we need to always be remembering that accompanying this is immune response as well. But this is a one-step growth curve that is typical for viruses and during a viral infection, especially in human beings. Okay, so we're going to have to think for a minute about viruses in bacteria. And that is for two reasons. First of all, we're studying microbiology. It's very important to realize that bacteria have their own many viruses which infect them. And there are different ways, there are two different ways in which viruses interact with bacteria. And the first is that they cause an immediate illness in the bacteria and a lysis of the bacterial cells. The cells actually burst. The second is a type of way that they interact is that they actually enter a phase where they are accommodated in the bacterial cell. They become latent for a long time and then are reactivated for one reason or another, and then go into a lytic phase. This is very important in maintenance of bacterial populations. Viruses are a major control mechanism for bacterial populations. And in fact, there are therapeutic ways of utilizing viruses to try and control bacterial populations that are causing an infection. But this is also analogous to some of the things that happen in human beings, for example, during viral infections. So it's useful for us to understand how viruses cause this lysis, or how they can actually become part of the functioning of the cell for a while and become latent. So that if they go into a phase where they cause immediate lysis, it's called a lytic phase, a lytic cycle. And the phage will cause immediate lysis and death of the host cell. A lysogenic cycle is different. In a lysogenic cycle, the phage genome becomes incorporated into the bacterial genome for a while, and it can last for a very long time. And then at some point or another, due to some stimulus or other, it will actually enter a specialized lytic phase it'll go into a lytic phase, as we'll see in a minute. So here is one of the typical bacterial viruses. This is T-even phage. There's a group of viruses called the T-even phages, of which T4 is one of the most famous. And I'll remind you, here's the capsid here, the geometric capsid. Here it has this long spring protein neck here. There is a tube which runs through there. These are filaments here that look like legs. These filaments guide the virus and allow it to bind to specific proteins on the cell wall of the bacterium. When it does so, these legs collapse. The spring, which is normally cocked open like this, collapses like that. And it sticks this tube all the way through the cell wall, which has got to get through, and through the cell membrane into the cytoplasm of the host. And it injects its DNA. There's the DNA strand being injected there. And we then proceed with viral replication, however it's. So here, these are important stages. And we'll see that these are the classic stages of infection for all viruses, not only the T-even viruses like T4. First of all, the virus has to attach to the host cell, to the correct host cell. In the case of the phages, they use these tail fibers. In the case of other viruses, they use those proteins on the exterior, either the exterior of the capsid or the exterior of the envelope, to find and bind to the correct marker proteins on the host cell. Phages penetrate. They do it by that elaborate mechanism. We'll see that in other viruses, they also penetrate in one way or another to get the genome into the host cell. In the case of these phages, that tube and that they punch through, it not only just mechanically goes through, but they actually have enzymes which break down the cell wall to allow to facilitate the penetration. Once inside the host cell, the viruses, whatever they are, they establish by one mechanism or another, take over the genetic machinery of the cell. And now, instead of transcribing the normal host messenger RNA and translating the normal host proteins, now instead the host cell begins to transcribe the genes concerned only with the virus, constructing viral proteins and replicating the viral genome. At some point, all of these different parts of the virus, the viral proteins, the viral genome, all the rest of it, these all have to be assembled inside the cell, and they are assembled and the nucleic acid is packaged carefully inside the capsid. Once that happens, then the virus has to escape. In the case of the lytic phases of bacterial infection by phages, the viruses actually produce a set of enzymes which will break through the cell membrane and break through the cell wall and cause the bacterial cell to burst. But other viruses may have other strategies for exit, as we'll see. But these T-even bacteriophages, they ultimately cause the lysis of the bacterial cell. So here's the lytic phase. Here's the virus, injects its DNA. That DNA, the first thing that happens is it directs the production of a set of enzymes which actually destroy the host genome, keeping only those parts of the host genome functional which are useful to the virus. It commandeers all of the transcription and translation machinery, and first of all gets the host cell to replicate the viral DNA over and over and over again, but then also transcribes genes which produce viral proteins. Those viral proteins, most of them self-assemble inside. Once they're produced, it is an inherent nature of the proteins that they self-assemble into the capsid and then the requisite parts of the virus. And the DNA, which is being replicated, packages itself into the capsid. Once that happens, viral enzymes cause the rupture of the cell membrane and of the cell wall, and the mature viral particles are expelled to go out and reinfect cells all the way around. So that's the basic scenario. The little complication that I'm going to mention now, because I'll refer to it again very briefly, that is, these infections are not necessarily always successful. Very often, when a phage injects its DNA into the bacterial cytoplasm, the bacterial cell has ways of recognizing that that genome is present, and they will cut it up. Their restriction enzymes are designed to cut viral DNA into pieces. And we now know that bacteria actually keep memory. They keep a record of the viruses that they have successfully vanquished. They keep pieces of their DNA, and they use it later on whenever they are infected. They refer back to this library, and they are able to respond to viruses rather in an analogous way to the way that our immune system keeps memory. It's very extraordinary. But you must realize what a huge danger these viruses pose to a bacterial population. This entire cycle in T4 in susceptible E. coli, for example, can go through this entire cycle within 20 minutes and explode the cells and explode out into the rest of the population, releasing hundreds, if not thousands, of viral particles. So it can go very quickly through an entire population of bacteria and lyse them all very quickly. So that's the lytic phase, where it just moves straight to go straight. The other viruses do not do this. They are able to go through a quiescent phase, and this is called the Lysogenic Cycle. And here, this is another bacteriophage. It looks almost similar. It's called Bacteriophage Lambda. And in Bacteriophage Lambda, instead of immediately cutting up the host genome and proceeding towards lysis, the viral genome, the viral DNA, integrates itself into the host genome, and it remains there in a form that is called a prophage. So all that's left of the virus, the only sign that the virus is present, is this DNA present in the host genome as a prophage. It still contains all of the necessary genes and everything to eventually produce phage particles. The point about this is that that is now part of the host genome. And as the host reproduces, divides, and increases its population size, every time this host cell divides, it replicates the viral genome as well. So the viral genome now, with no further intervention by the virus, begins to spread through the population. As the population increases and clones itself, it also clones the viral genes that are present in the genome. And this means that now the host is actually like a hybrid between the genome is a hybrid. It has bacterial DNA genes, but it also contains the viral DNA. But it does not necessarily remain that way forever, because the lysogenic phase, the bacterium reproduces over and over and over again. It reproduces the integrated viral genome over and over.